Caleb Burruss, and this is February 13th, 2023. So to start off here, I have a cell, and I have all the main organelles we're going to talk about within these three macromolecule movements. So we have the cell membrane, we have the lysosome, we have the Golgi apparatus, we have these little red dots, which are our ribosomes. We have our mitochondria here in purple. We have our smooth ER here in yellow, our rough ER in orange, and then our nucleus here in this little pinkish red. So our first molecule we're going to talk about is the steroid hormone, or hormones. So steroid hormones start out in the smooth ER, and we start out with their precursor, which is cholesterol. So cholesterol is formed in the smooth ER, and that is going to be put into a transport vesicle. And this transport vesicle is going to transfer over into the Golgi apparatus. More specifically, it's going to travel into the cis side of our Golgi apparatus. And then it'll come out the trans side, go into another transport vesicle, and then finally be shipped off into the mitochondria to do some changing. So once it's outside the mitochondria here, we're going to hit our outer mitochondrial membrane, which is the dark green, and then we'll be transferred into the light green, which is the inner mitochondrial membrane. And within this, we have our cholesterol. So now our cholesterol is in our inner mitochondrial membrane. And what's going to happen next is it's going to be changed into pregnenolone. Okay, and once it's in pregnenolone, it is going to finally be shipped back out into another transport vesicle. Okay, and so this pregnenolone is our precursor, our main precursor for most all steroid hormones. So both these two, cholesterol and our pregnenolone, are precursors for our steroid hormones. So once this is kicked back out into a transport vesicle, it is going to travel back into the smooth ER. And once it's in there, it is finally turned into a steroid hormone, which is turned into a secretory vesicle. And so once it's in this, it is going to move to the cell membrane, where it is going to be secreted out by exocytosis into the extracellular matrix here, and it is going to be moved out to wherever it needs to be within the body. This could be bloodstream, it could be muscles, it could be straight into other tissues. It could be a lot of different things. But one we're going to focus on today is going to be cortisol. So cortisol is made within the adrenal glands, so down here on top of our kidneys, and it is secreted straight into our bloodstream. So cortisol here is in green, and it is secreted right into our bloodstream. And then once it's in our bloodstream, it can go to many different places. So I chose just three here, for example. But we use cortisol. Okay, so the reason that this is being formed, right? This is formed just like we just talked about on the previous part of this flow diagram. We talked about how it is made in the cell, and this is a steroid hormone. And so the reason we make this steroid hormone is because of stress. So when any times of stress, this could be physical harm, like if you're under attack or in danger, your body's going to secrete a whole lot of cortisol, which is going to go into the bloodstream, and then it's going to travel to a lot of different places. So it may just stay within the bloodstream, okay? And this is going to increase your blood pressure by vasoconstriction. And then our heart rate will increase, so that's going to cause more blood. It's also going to cause higher blood pressure. And then it can also go to the brain, because the brain tell the brain that it needs to use increased glucose consumption, so that you are staying on top of your wits and you're using your brain at full power here. Our next type of hormone is the peptide hormone or protein hormone. So peptide hormones, instead of being started in the smooth ER, we're going to start in the rough ER. And so we get our information on how we're going to code or rate these different peptide hormones from our nucleus. So instead of starting with cholesterol here, and then transferring to the Golgi apparatus, and then back to mitochondria, and then out of mitochondria back to the smooth ER, like we did with steroids. With peptide, we get all information straight from the nucleus saying, okay, here's our mRNA. It's going to be delivered to one of our ribosomes, and this ribosome is going to create our peptide hormones, and that's going to be kicked out into a transport vesicle. And so now this transport vesicle is going to go to the Golgi apparatus, because the Golgi apparatus is going to tell this peptide hormone where they're going. So we'll talk about a couple different places where it could go. And so it's going to kick out the trans side, and it could be put into a secretory vesicle. And this is what makes it a peptide hormone. And so There's two places it could go here. It could go into a lysosome. We'll talk about how the intracellular proteins can also do a similar thing, or it will be kicked out of the cell, similar to a steroid hormone by exocytosis, and it's kicked out into it could be bloodstream, could be tissues again, lots of different things. So one peptide hormone we're going to talk about is insulin. And so here with insulin, we have in our green little triangle here, it's going to come and it's going to find a cell with an insulin receptor. It's going to hit that insulin receptor, and it's going to cause a cascade of secondary messengers within the cell that will tell the cell, okay, insulin just bound to its receptor. We need to pick up some more glucose, and so it's going to open up these protein channels within the cell membrane, which is going to allow more glucose into the cell. And then finally, we have intracellular proteins. So with intracellular proteins, same thing as peptide hormones. We're going to have mRNA that comes here. It's going to bind to our ribosome on our rough ER. It's going to, or it could be, let me add to that, it could be a ribosome within the cytoplasm here. It doesn't have to be on the rough ER. Usually it will be. And so it's going to come on here. It's going to be made into a protein. And then instead of going out of the cell, it can go to a couple of different places. So we put in this transport vesicle. It could go straight to a lysosome, just like peptide hormones. So we want to send these to the lysosome because a lot of proteins are used to help break down chemicals or anything that, I guess you would call it trash in the cell. So organelles that have died off or things like that, to get rid of them. It could go into the Golgi apparatus and help. There's proteins in the Golgi apparatus to help tell things where to go. So it could help tell where other peptide hormones to go. It could tell anything that the Golgi apparatus needs to send off to somewhere else in the cell, then it could go through here. And then it could also be sent off into the cell membrane, okay, and become a membrane protein. So there's a lot of different types of membrane proteins. This one I showed here is more of a protein channel that is made here, and there's lots of different types of those as well. And so it may embed itself in here and kind of do its job within our cell membrane. And then something else I haven't shown here, because I'm going to talk about on the next slide, is that it may go back into the nucleus. And that's something we call, or one example of that is DNA polymerase. Okay, and so we get mRNA that comes in to a ribosome, and then it's going to go back into the cell, into the nucleus. And then in this little wispy stuff here is the DNA, and it's going to copy the DNA in times of cell division. Okay, and so it's just one of the many examples of intracellular proteins that we'll see within the body, or even within the cell, I guess, in this case.